Welcome everyone to CTS's ICU Surge Weekly Meeting. Christian Sandrock and Danny Amundson and I have been doing this since early January so that we can stay out of surge. And we welcome all of you tonight to our sixth or seventh meeting. And we're going to be talking about the monoclonals, the variants, then vaccines. And welcome. Before we get started, just wanted to acknowledge Fisher Paykel, SmartVest, and Voxen or Ventec for supporting us for this platform. And of course, the folks behind the scenes at CTS, Vicky, who has been our ringleader in coordinating all of this, and Phil Port, and Dave Eubanks. So before we dig into the program, here's just a little sit rep on what is currently going on in our state. As of just a few minutes ago, I downloaded this from the State of California website. And it's important to note that, well, here they're saying that we've got an increase from the day prior in deaths, but the arrows are going downwards. And that's, I think the arrows are incorrect. The trends are still going down, and that's what they want to highlight. However, if we look at our ICU beds available, we're still right where we were at in December. So we still have a long ways to go for bed availability. And if we look at, I just quickly pulled out San Diego's, and I mean, we only have about 50 beds available right now. And we average usually anytime before COVID at about a 70% occupancy. And during flu season, if there was an active flu season, then we are a little bit higher at 80 to 90%, but we still are busy, and as all of you are busy. And so with that, we'll dive into our agenda. So we'll start with our usual surge updates or ICU updates from across the state, throughout the state, followed by Shazia Jamil discussing the genetic variants of COVID-19, and Ryan Maves from Navy Medical Center San Diego will discuss COVID-19 vaccines. And we are honored to have Dr. Charles Delacruz from Yale to talk about the monoclonal antibodies in the treatment of COVID-19. With that, Christian, I'm going to toss the ball to you, and we'll try to run the board. I'm going to open the chat box to see who might be on board so we can get a sit rep from folks on the ground.
Yeah, sounds great. So Denny is tied up in the ICU, which hopefully is not a bad prognostic feature for how his ICU is doing. So he can't give the update from Southern California, but we'll rely on anybody else from San Diego, Los Angeles, or Fresno if you're on. The quick update for the Northern California, and probably the best thing, is that not much has changed. It's been a relatively boring week, and we will definitely take boring. Our numbers are pretty much flat and consistent with what Asha showed, where there are still some availability. We at Davis really haven't had any, but we've been able to squeeze out a little room for some transfers. Our ECLS starts and terminations have been flat, so we really pretty much have had the same number of people on circuit. Our total number of ventilators in use has also been flat, although there's been a little switch away from a few of our COVID people to get some non-COVIDs on mechanical ventilation, but our ICU beds and mechanical ventilation use has also been relatively flat. So that is consistent with most of the greater Sacramento area. And from the little bit I got from our MOAC, that's pretty consistent down in the Bay Area as well. Although if anybody from UCSF or elsewhere wants to chime in, please feel free. And then we'll see if there's any updates from anybody in Southern California, and we can open it up before we really the best smart thing is to keep my mouth shut because we have some great presenters tonight. So we'll give a quick update and give them lots of time to talk. So
I see some folks from Southern California on the line, Vipul, or Central California. Vipul, do you want to tell us how's Fresno? I know you guys have been hit hard.
Yeah, actually, fortunately, I have the same kind of census trajectory, like was mentioned. We were slowing down last Friday. Like I said, there was a couple nights we were busy, but we have overall been on a declining trend. So our ECMOs are flat, no new ECMOs. We have not seen any major bursts of COVID from Super Bowl or anything yet anyway, or variants. So fortunately, I'm hoping, the fingers are crossed. We had put 3 extra teams for the surge. I've aborted 2, and I think this weekend or Monday, the only extra team that's in place right now, I'll probably put it on standby. So yeah, we're really key. And transfers, the other hospitals, we have gotten much less requests for transfers now. We are able to accommodate some of the transfers because, again, we have these extra teams in place. But compared to a few weeks ago, where we were really declining any transfers, we're in much better shape. So hopefully things will continue the same.
Anyone else from Southern California or Northern California who wants to give an update?
Yeah, Scott McCall from San Diego, La Jolla.
Perfect.
We're very, very similar experience. We're opening up more beds, doing more cardiothoracic surgeries, still getting, the occasional referrals. We're at 9 in our institution. And as far as the county is concerned, we have like a couple slots to go on ECMO and have still suspended ELCRs. So that's at the discretion of the ED doctor. We still get calls to weigh in on folks being transferred in, but it has to be a witnessed arrest within our ED, somebody too young to die sort of situation.
This has been some timely stuff. If you guys have been watching, obviously, as Pfizer released a little bit of data with the B.1.351, and there's some new data on the P1 variant, and it'll be great to see with both vaccinations the impact of the variants and whether we can project what our census may be as these variants take over. So perfect. Well, with that, I'll keep quiet. Asha, if you want to introduce, I'll defer to you, and this is going to be good.
Yes, thanks, Christian. Well, Shazia practically needs no introduction. She's a pulmonary critical care and sleep specialist at Scripps in San Diego and has a ton of experience in titles, especially ATS's rapid response document team, and is working on a guide for ATS regarding the genetic variants. And she's going to share with us some information right now. So Shazia, the floor is yours.
Good evening, everyone. Can you hear me, Asha and everyone else? And can you see my?
Yes.
Thank you so very much for inviting. This is a very exciting time and exciting speakers today. So I want to go ahead and start my presentation. Why is it not moving? Excuse me. Yes, I do not have any conflict of interest, and due to the evolving nature of this presentation and the topic, I must say that if another variant has come up since 12 noon today, I do not take responsibility, but we'll talk about it maybe later soon. Okay, so I just wanted to introduce my team. I've been very, very lucky to be colleagues and working with a group of American Thoracic Society clinical educators, public health workers, as well as physician scientists. All of us are clinicians and also clinical educators. And about 5 years ago, just after the education committee meeting in New York, we were sitting down and thinking about how rapidly disseminate information. At that time, we were experiencing Hurricane Harvey, and this is how this committee or a small group came about. And we actually do take a lot of pride that we started working on the now SARS-CoV-2 virus, at that time called as Wuhan novel coronavirus. Really in January, before the first case got identified in the United States, we actually collaborated and worked with Dr. Ben Cao, who at that time was president-elect of the Chinese Thoracic Society. And our paper, I think, was trading under 10 on PubMed and online published of February 1st. And since then, we now have about 6 publications. And our goal is to basically create an expedited evidence-based short and practical document to not only educate our healthcare workers, but also our patients throughout our country as well as the world. So, and Charles Delacroix, who would be talking later, we have invited him, who has been instrumental in this group. I must say that my presentation is going to be a lot technical. I'll take my time to explain things, but as you can see, whether you turn on news or you read a newspaper article or pick up a Beat or JAMA article, you would come across a lot of technical terms when we are talking about genetic variants. And some of the questions that come to our mind, which are very practical questions that as a clinician, are the following. Is the RT-PCR technology that we use, and that's usually the most common technology being used in the world and in the United States, is it able to detect some forms of mutation? Are these mutations more transmissible, more virulent, more pathogenic, or more deadly? How about if we already had infection and now we're recovered? Are we more likely to get the variant or will be immune due to it? How about convalescent plasma and its role if for example, we are infected with the variant? How about vaccine? And more importantly is the monoantibodies, which are more in the news and in the articles that we have been published and getting emergency use authorization. Would they be of any use if a patient is diagnosed with the variant? And finally, are children and young adults now more susceptible because we have variant compared to before? And what about the public health measures? Are they enough, what we are doing? And in the hospital or taking care of the patients, do we have current PPE measures enough to prevent spread in hospital setting? I will try to briefly address each one of these questions, which I think are very practical. Again, this talk is going to be a little bit technical, and I'll try to be as simple as possible, but an editorial that got published by Dr. Fauci just recently, February 11th in JAMA, actually stressed to the clinician to be aware and have understanding of SARS-CoV-2 genome as well as this terminology. Otherwise, we will all be very, very confused. So I tried to put in this slide very simple definitions. So mutations could be a single nucleotide mutation, such as the one that we had seen earlier on, such as January and February mutation in 2020. And I will describe that. That is now actually circulating as more than 98% of all SARS-CoV-2 virus, which is called the D614G. A variant is, as is described in this slide, when there are changes in the RNA due to mutations, then there is a variant. However, variants can all be very, very similar, and until the phenotype is changed. So the virus is behaving a little bit different, for example, more transmissible, more deadly, or affecting a different age group or whatnot, then only we will call it a strain. Lineage, you will hear a lot, for example, B117, B135, etc. So what is lineage? So lineage is a little bit more complicated, but you have to actually go through the phylogenetic tree to figure it out. And just remember that the SARS-CoV-2 belongs to the beta coronavirus, and these are lineage B, most of them. And that's why B is in front of many variants' name. Finally, clade. I mean, this is something also we are actually reading in the articles that are coming out. And this is various ways species and subspecies relate to each other and can be used to track how viruses bounce all around different geographical areas. Traditionally, we believe that SARS-CoV-2 has 7 genetic clades. However, more recently, a group of really epidemiologists and scientists have created this public forum by the name of Next Strain. You can go on right here, which is available online, and go and look at this group. And they have actually now trying to identify and change the clade nomenclature. So for example, you may see 19A, which actually is the first clade from Wuhan, year 2019, and 20A is year 2020, first time diagnosed in Europe. quite complicated, but very, very fascinating to me, looking at the phylogenetic tree and how from, for example, the European 2020, when it got detected, from there, these are the descendants. And now we're talking about the 501Y, which I'm going to describe a little bit further. So just so that you understand why there are so many different numbers in front of these genetic variants. So the current naming of SARS-CoV-2 is extremely confusing to everyone, and especially practitioners, because we're not really used to, the only thing we are used to is the influenza virus. We talk about H1N1 or H1N3. So 6 years ago, the WHO introduced guidelines to end naming of viral diseases. They do not want us to think about landscapes, regions, and cultures, for example, Middle East respiratory virus or Zika virus. Zika is a forest in Uganda, for example. They don't want us to do that. However, those guidelines only apply to new infections, but not the variant. So we are back to square one. So right now what's happening is that the coronavirus variants are being named again, unfortunately, according to the geographical locations where the first cases were identified, and then sometimes by the scientists who have discovered these variants, they would try to be more scientific. For example, the South Africa variant is also called the 501Y version 2, which is a substitution of the 501st amino acid site in the spike protein. On the other hand, the UK variant, a little bit different terminology, is the variant of concern. 2020 is the year, 12th is December, and is the variant 1. But it's also being classified, it's exactly the same virus being classified based on lineage. So it's B117. So it is definitely confusing, but it's important for us still to know what is going on in the literature. So there was an urgent meeting of WHO in January of 2021. And at this time, the last time I checked, as of today, they have not yet come up with a new nomenclature. So again, it's a little bit technical presentation, but it is so very important that we understand that, like even literally in the news, public is being notified of N501. I was reading an article in New York Times and it talked about it. So yes, it is technical, but we really need to know. So very simple diagram of a spike. The spike actually needs 3 spike proteins to make 1 spike. And these are the most common mutations that you will hear again and again and again. Being an RNA virus is actually really common for it to undergo mutation. So what is the 501Y, which is really making a lot of news? It actually helps virus bind more tightly to human cells while ACE2 receptors. Therefore, when this mutation happens, it is more transmissible. How about the E484K? Again, single point mutation in the nucleotide, and it helps virus evade some antibodies. This is the new mutation, which has been now confirmed as at least 45% of all samples tested in California, and especially in Southern California, has been found. And finally, this is the most latest, is the Q677. Q actually is for glutamine substitution. And this is very important because it's located at the cleavage site. And scientists believe that because it changes the conformation of the spike protein and it opens it up, it actually binds much more effectively to ACE2. And finally, our ancestral mutation, which is now, is circulating more than 98% of the entire SARS-CoV-2 in the world, was the first single point mutation, D614G. Let's talk a little bit about the D614G mutation. So as I mentioned, it actually started circulating very, very early. Remember that at that time, the novel China Wuhan virus was circulating in November, December in China. So finally in January, February in Eastern China was the first time that this mutation got identified. And it's definitely in the S protein. It actually probably is the reason why the pandemic went so fast, so early. And we believe that all the other variants are descendant from this. It also affects and binds ACE2 very, very efficiently. These are some very cool experiments that are done and were published in Nature by Dr. Plante. And it actually shows that both in the cell culture in the human airway epithelial cells compared to the wild type, which was the previous SARS-2 coronavirus, this mutation actually really replicates more, it binds more, it is found at a higher nasopharyngeal load. But one good news is that when they used the neutralization assay for this mutation, they actually found that the antibodies were binding, the antibodies which were against the initial non-mutated D6114 were binding to the now mutated 614G. So that, I think, is a very important good news. So I think one important question that always came to my mind when I was listening to all these mutants and being an ICU physician is when we are sending the RT-PCR, are we detecting the SARS-CoV-2 in the presence of mutation or the variant? So let's think about what really happens in the when we order a PCR. So there is a selection of target regions for PCR primers, not just for SARS-CoV-2, for any other viruses. They have to be specific to the virus. They have to be directed to the highly conserved regions where the mutations might be less frequent. So again, a little bit technical, but I think we again need to know some of this stuff that this is basically what is in the cartoon form, your SARS-CoV-2 virus. It has spike, it has the RNA. Now, you can see that the RNA codes this amount, a small amount of RNA actually codes the entire spike protein. In terms of essential proteins, we have the spike protein, the envelope, the membrane, and the nucleocapsid protein. And then we have some non-essential proteins that are actually translated by ORF1, which are the open reading frame proteins. A little bit technical, I apologize, but I thought it is important for you to know. So let's take it, for example, the Thermo Fisher assay. That detects 3 targets, the S protein, the nucleocapsid protein, and the ORF1 gene. If you send a test, which actually has the Thermo Fisher, it's a Thermo Fisher RT-PCR, a positive assay will be reported if you have 2 out of 3 genes detected. And when there is a drop in the S gene, that should be suspicious as to why this assay did not detect the S, protein. So I hope it makes sense. So when it's suspicious, then we should do some genomic sequencing to figure out why this test did not detect the S protein. And some other example I just wanted to put out, there's actually now multiple, both commercial and other kind of RT-PCR tests. But as you can see, almost all of them actually take into consideration more than 1 genes. So the primer should bind to 2 different, regions in the gene. So just to give you an example. So then what happens? So we should be utilizing assays that targets multiple targets, and that way it would mitigate the risk associated with mutations altering primer binding. And as I said, it has to be confirmed by viral genomic sequencing and not just by RT-PCR. And at this time, all these commercial companies are very, very closely looking at the genomic variants, trying to change their primer binding as we speak. And at this time, we believe that RT-PCR by any of these tests can pick up SARS-CoV-2, even with variants. So let's talk a little bit about the B117 lineage that has really shaken the world, and it's now found in more than 70 countries and it's growing. So as I described before, it has multiple names. Sometimes you will hear this or the WOC 2012/01, or based on the single point mutation for 01Y, it may be called this variant. It has many mutations rather than just one single nucleotide mutation, and it has 8 mutation in the spike protein. And then if you tease it out, the most important mutations, as we just talked about previously, the 501Y, but in addition, there are other mutations which are actually making this virus very, very transmissible, we believe, up to 30 to 80% more transmissible, that contributed to UK government decision to enter a national lockdown. There's a possibility of infecting all age groups in a similar way. And the reason I say possibilities with caution, because as I put out these references, many of these references have actually not been peer reviewed. However, the data was there and it actually still led the UK government to take measures. It is doubling in the US every 10 days. And again, based on some data, it could be 35% more deadly than other variants. As you could see in the map of the United States, it's really widespread. And this was downloaded a few days ago. Who knows now, maybe some other states will be involved. So what about the B1351 lineage? This is, as you may have heard, the South African lineage. It also has 3 most important mutations in the spike protein. It is also highly transmissible. However, at this time, we do not have any data regarding risk of severity or death. Possibly, again, based on the preprint articles that are out there, and the governments and CDC are also looking the same articles and actually have been referencing these articles as well, maybe less effectively neutralized by convalescent plasma or by sera from those vaccinated, maybe resistant to certain monoantibodies that are being used. And I think this may be very, very important, that those recovered from other variants may not be immune to this variant. But both Ryan and Charles will be going through this definitely in much more detail. But I just wanted to provide a framework as the first presenter to make sure that everyone understand what we are talking about when we talk about mutants and these variants. At this time, not that much widespread compared to the previous one, but definitely increasing. So then you may have heard about the Brazilian strain. It's actually very interesting that 4 people from Japan went to travel to Brazil. They came back and the National Institute of Japan somehow picked their sequences. And then later, it's now 42% or even more specimens from especially a part of the Amazon region has this variant, extremely fast growing in Brazil. It has also a lot of mutations, but then the 3 mutations that we talked about previously, this variant also has these mutations. Diagnosed or detected in the United States, January 2021 in 2 states. Pretty sure, again, this was downloaded a couple of days ago, may have further expanded. It is also highly transmissible and concerns of a potential propensity for reinfection for individuals. So since we are in Southern California, this report came out from CDERS. It came out February 11th. They actually looked at some samples that were already collected from 2019, December to January 2021, and they found 5 different mutations. All of them seems like really at least three of them seems to be in the spike protein and other genetic codes. And they designated this as Cal20C. This is making a lot of news, much more in Southern California, as you can see, but throughout the California. And this paper actually led us to this right here, because they talk a lot about the L45-2R mutation, single point mutation, first identified actually in Denmark and not in the United States, but somehow has now traveled to the United States. It could also affect binding of certain monoantibodies. It has really progressively from 3.8% to 45% or more of current California samples, and then has really rapidly spread to 26 states. And this is the article if you would like to read a little bit more about it. So then, as I thought that I was finished with my presentation just a few days ago, guess what? Another really excellent scientific discovery from a collaboration of University of New Mexico and Louisiana State University. This was posted February 14, 2021. Many journals are actually talking about it. So they are calling these yet another set of mutations which they have found in Q677 lineages. Q actually stands for glutamine, if I remember correctly. And they're actually multiple variants. So there are probably 7 new variants. And now these names, I mean, go figure, kind of a thing, Robin 1, Robin 2, pelican, yellowhammer, mockingbird, bluebird, quail. Well, we will have to remember all these names. But in any case, so this is again preprint, but this is making a lot of headlines right now, and we need to know about it. It's present in the world. It's also present in the US. It's relatively, it seems like not really in California that much, but you know, a lot of people are traveling. So who knows, we're gonna see that. So this is all for variants. And then I think most important thing is how can we prevent infecting from variants? And I would say that it is much more critical now than before to implement strict public health measures by all, whether we are vaccinated or not. And this sentence, if you can remember, I think it's important that viruses do not mutate unless they replicate. And everything we do, we are trying to avoid viruses to replicate. So I think both in the hospital, in the clinic setting, as well as outside our homes, we have to, if anything, much more cautious now than before. And with that, I would leave you with this slide. What does the future hold? So will selective pressures of expanding herd immunity, vaccine-induced immunity, antiviral therapies, and public health intervention strategies would drive antigenic variation, would promote virus stability and transmissibility, would alter virus virulence and pathogenesis, or it would drive SARS-CoV-2 to extinction, or would SARS-CoV-2 would find alternate hosts as reservoir. And this is from an editorial written by Dr. Barrick in New England Journal of Medicine. It's an excellent 2-page editorial, and I would like everyone to read if they can get hold of really perspective about where we're going in this world and in the United States. I really thank you for your time, and thank you for listening to a very technical presentation.
Well, thank you, Shazia. That was excellent. And in the interest of time, we'll just segue right to Ryan and then Dr. Dela Cruz. And if anybody has questions, please use the chat box function. So with that, I'd like to welcome Dr. Ryan Maves from Naval Medical Center, San Diego, to give us an update on the COVID-19 vaccines, efficacy, maybe the impact of the variant strains. Ryan, the floor is yours.
Thank you very much, Asha. And following Shazia is always a challenge. While I'm getting my slides, I want to say that we're taking a vote and we're making Shazia an honorary ID doctor after that tour de force of viral phylogenetics. So thank you very much. All right, so my name is Ryan Maves. I'm an infectious disease and critical care physician at the Naval Medical Center. I also chair the ACCP's COVID-19 task force. For disclaimers, I have no relevant financial conflicts of interest, but I am, however, the site principal investigator in San Diego through the Navy for the AstraZeneca Phase 3 trial. I'm also the site PI and DoD coordinating PI for the NIAID adaptive COVID-19 treatment trial. These are my opinions. They're not the opinions of my employer or the government. There will be some discussion of off-label and investigational agents. All right, so just kind of leading into this, the challenge, and I think all of us know this after a year, about looking into the data underlying things that we do for COVID-19, and vaccines are no exception, is, we have this traditional pyramid of medical evidence where kind of at the bottom is expert reviews, and you go up to RCTs, and some people would say systematic reviews and meta-analyses are the pinnacle of medical evidence, although I think there's another school of thought that would say that the RCT is the pinnacle and systematic reviews serve another purpose. What we have nowadays is more like this. where we are dealing with press releases and preprints and non-peer reviewed data, and we are forced to make both clinical and policy decisions based on that. I will mention that the only two parts of this slide that are not meant to be a joke are Fauci's head on top and RCTs right below. So just a quick snapshot of how quickly we're vaccinating people. So the Biden administration announced upon taking office that the goal would be to get 100 million vaccine doses given in 100 days. And interestingly, so far, the average rate of vaccination in the United States has actually exceeded that relatively conservative target. So far, we're averaging, as of the New York Times last night, 1.62 million doses given per day. The country that leads the world in terms of the proportion of their population vaccinated is Israel. United States comes in number 5, right behind the UK. So I guess you can say we're leading in our weight class, but we certainly have a long ways to go before we have effectively vaccinated the majority of our population. So, okay, there we go. All right, so when we look at vaccine development, the challenges we've had in developing effective vaccines for SARS-CoV-2 is essentially one, that we've never had a human coronavirus vaccine before. There are animal coronavirus vaccines that have been in clinical use by veterinarians for some time. So the proof of concept is already there. But the other challenge is this: there are two relevant arms to the adaptive immune system. We have to target both antibodies, meaning the humoral immune response, as well as a cell-mediated response. Traditionally, the main way we've been able to elicit cell-mediated immunity has been through an attenuated live virus vaccine. The problem, of course, is that many people, including large proportions of the groups at greatest risk for severe SARS-CoV-2 infection, are generally considered ineligible for live virus vaccination. The very elderly, immunocompromised patients, pregnant women, and the like. So all of these new technologies are ways to get around that problem, to induce not only humoral, but also cell-mediated immunity without having to give a live virus. So nucleic acid vaccines, the mRNA vaccines that are the two that are in use right now, the Pfizer and BioNTech vaccine and the Moderna vaccine, are one way around this. There is some ability to scale them. They can be produced relatively rapidly, but they are expensive to produce, and that can be limiting from a global health standpoint. Viral vector vaccines, this includes the AstraZeneca vaccine, the Johnson & Johnson vaccine. Merck looks like they're kind of shelving their current vaccine candidate. So these use a viral vector, generally an adenovirus, that is modified to encode for the SARS-CoV-2 spike protein. They are modified additionally to be replication incompetent, meaning they can enter your cell, they can interact with antigen presenting cells, interact with major histocompatibility complexes, generate a cell-mediated response, but they will not replicate inside of a mammalian cell. So this gets around some of the issues with the immunoincompetent host. There is some theoretical safety concern for replication despite this, although this has not thus far been seen in randomized trials. Admittedly, relatively small numbers of pregnant women have been vaccinated so far, and relatively small numbers of pretty healthy HIV-positive folks have been vaccinated so far. Inactivated vaccines are in development by Sinovac. That's being tested in Brazil fairly heavily right now, and a number of other largely Asian companies. These have the advantage of being cheap, easy to scale. They've been licensed before. The downside is you get only humoral immunity and no cell-mediated immunity from that. There is this theoretical concern of sort of vaccine-related enhancement of the inflammatory process. This is, for those of you familiar with how dengue hemorrhagic fever works, how you have a heterotypic dengue infection leading to more severe disease. This is the same sort of concept that hasn't really been observed with human coronavirus infections, but it has been seen in feline coronavirus infections. So it is a possible concern. Then lastly, protein-based vaccines. So Novavax is the one that's furthest along. Sanofi Pasteur is developing one. The US military, the Walter Reed Army Institute of Research is developing this one as well. And I should probably say that the Walter Reed Army Institute of Research is where I got this particular slide from, courtesy of Dr. Mo Jarrad at the RARE. All right, so what are some of our safety pitfalls that have come into concern? Now, fortunately, what we've seen thus far in vaccine development is we haven't had a lot of these safety pitfalls. And although all the vaccines that are in late development are very reactogenic, meaning that febrile reactions, malaise, headache, these are relatively common after the Pfizer and Moderna vaccine. They seem to be maybe a little less common with Astra, but not that much. But in terms of genuine safety threats, other than infrequent cases, thankfully very infrequent cases of anaphylaxis, but well described, these vaccines appear to be pretty safe. But there is always the concern about, with some of these new technologies, could we, for example, induce immune complex formation and complement deposition? Could we induce complement activation and a hypersensitivity reaction? How would these vaccines interact with prior beta coronavirus infections, the common cold viral infections that most of us have had in childhood? So these are the concerns that come up, but thus far, these haven't proven to be significant as we've gone through phase 1, phase 2, and now phase 3 trials. All right, so let's start by taking a look at nucleic acid vaccines. So the idea, so DNA vaccines are on here, but really what we're looking at is RNA vaccines, and specifically messenger RNA. So the idea is you would coat messenger RNA that encodes just for the gene sequence of the SARS-CoV-2 spike protein. The lipid coat is so that it can enter cells. These are injected into the human host, taken up into the cell, enter your nucleus. Do not enter the nucleus. That's what DNA vaccines do. mRNA vaccines in the cytosol interact with human ribosomes to produce viral spike protein. Then your myocytes, it's generally going to be skeletal muscle cells, produce spike protein interacting with the major histocompatibility complex and leading to an immune response, both antibodies and T-cell based responses. So this is a technology that's been in development for a very long time. Actually, the US Navy had a dengue vaccine, a dengue mRNA vaccine that was in late development. It was not successful, but that's more due to the vagaries of dengue immunology than anything else. And the Zika mRNA vaccine probably would have worked, except that Zika has largely faded, although there's, interestingly, I was reading the paper yesterday, a possible upcoming Zika outbreak. So Pfizer and Moderna. So the left-hand side of this slide is the Moderna vaccine data from their phase 3 trial in the New England Journal late last year, the mRNA 1273, that's the Moderna vaccine. And this is just showing incidence rates over 120 days of follow-up in patients who received either Moderna vaccine, that's the blue line, or placebo, that's the dark gray line. And you can see 94.1% efficacy in reduction in all-cause cases of SARS-CoV-2 infection, and an even greater reduction in the incidence of severe disease requiring hospitalization. Very, very similarly on the right is the Pfizer data. It's a BNT162b2. Those of us who do HIV medicine are very used to long letter number code names for drugs before they get real names, but this is so BNT is the Pfizer BioNTech vaccine. And here you can also see that there was a 94.8 reduction that kicked in 7 days after the second dose, right? So these are both multi-dose vaccines. And I suspect a great many of us on this call have received our vaccines for these due to our lines of work. So extremely effective and honestly better than any of us would have expected. The initial target from Operation Warp Speed, before it was called before its name got changed, was 50% efficacy, which would have been, a pretty good flu shot. And instead, we get numbers like this from the mRNA vaccine trials, which gets us into measles vaccine territory, at least in terms of short-term efficacy. So what are our concerns? Because this is obviously a talk not just about the vaccines. We're all probably reasonably familiar with the Pfizer and Moderna vaccines now, but really about what are our concerns going forward? So right now, the big concern about the variants, specifically the UK variant, the 117, and then the 1351 variant from, 1135, killing me today. 135 variant from South Africa. So here on the right-hand side of this slide are comparisons in neutralizing antibody levels from vaccinated hosts when exposed in vitro to both kind of what we'll call wild type SARS-CoV-2. This is the top one is a specimen drawn obtained in Seattle very early in the pandemic in the Pacific Northwest. And you can see that there's kind of no real change between this and another variant that's been circulating more commonly. But then when you compare the levels of neutralizing antibodies in the slide below to the B1351 spike protein mutant that we're discussing from South Africa, that there is a significant decline in the levels of neutralizing antibodies in these hosts. So about a 2/3 reduction in general. These are geometric means, so it's not a completely linear relationship. But so the question then becomes, what does that mean? What is our threshold for immunity? Because no vaccine is going to function by inducing the bare minimum amount of neutralizing antibody. So is 1/3 of that baseline, is that enough to convey protection? The other open question is, of course, that these vaccines also induce a CD4 and CD8 response. What is the impact of these mutants on that response and on that memory response? So we really don't know what level of clinical protection these vaccines induce. We do know that antibodies produced by them have decreased tropism against the South Africa variant, against the 351, but they still have some activity. And the question is that activity enough? And we simply don't know at this point. The Moderna vaccine, the mRNA 1273, has a similar decrease. It is apparently still active in vitro, but again, with a comparable decrease in neutralizing antibody titers. So how this will play out remains unknown, but it does appear that there is going to be at least some degree of protection from the mRNA vaccines. Another kind of open question, which is interesting, this was a letter to the editor in the New England Journal by a Canadian group who pointed out that the Pfizer vaccine, the BioNTech vaccine, had a 92% efficacy after the first dose. And in times of vaccine shortage, is that extra 3% of protection worth it? Should we be switching to a single dose regimen? Now, right now, the FDA's response to that is no, this was the 2-dose regimen is the validated dosing regimen, and that's one we're going to stick with. But it is an interesting thought for us when we decide what to do as a society going forward, is if in times of vaccine shortage, should we just give one? Right. So anyways, an open and interesting question. Alongside with this, there is a series that has not yet been published by the leading health maintenance organization in Israel, again, which has vaccinated a very large proportion of its population, including over 75% of their at-risk population. And what they have seen is that in a real world setting, comparing 600,000 vaccinated to 600,000 unvaccinated Israelis, that this 95% protection appears to be playing out in the real world. And that's very encouraging as our vaccine programs move forward. All right, now how about viral vector vaccines? So the idea behind it, again, most of the ones that we are looking at here are non-replicating viral vectors. So the AstraZeneca vaccine, and we'll also talk about the Johnson & Johnson vaccine. So you have a spike protein gene from the coronavirus inserted into the adenovirus, which then enters, undergoes processing inside of the human host, but does not replicate. All right, so right now, in terms of the kind of formal peer-reviewed literature, we don't have a ton of data that's actually made it to publication clinically on the ChAdOx1 vaccine. So the AstraZeneca vaccine is called the ChAdOx1. That stands for chimpanzee adenovirus Oxford 1. That's the viral vector. The AstraZeneca vaccine uses a chimpanzee adenovirus, which was chosen because of concerns with human adenoviruses, that a lot of people will have antibodies to human adenoviruses and thus may not respond to the vaccine appropriately if a human adenovirus vector is used. This has been seen in other vaccine trials, most notably and recently Ebola. Chimpanzee adenoviruses are perfectly capable of infecting human cells, but humans generally do not have prior exposure to chimpanzee adenoviruses. So this is the phase 1. This is a collection of phase 1 and phase 2 trials conducted in Brazil, South Africa, and the UK. It's worth noting that these were conducted before the emergence of the 351 variant in South Africa, however. So that's going to affect a good bit of what we know about this. So overall, what they're reporting in these phase 1, phase 2 trials, relatively small studies, is about a 70% efficacy in these populations. The problem is that these are safety populations, right? So these are generally people who are 18 to 55 years of age, relatively low risk. They are overwhelmingly white. There are about 65% women in these trials. Still plenty of men to get you evidence of efficacy. One of the interesting things about this, and this was actually done, I think it's actually modestly embarrassing for AstraZeneca, to be honest, but a large proportion of people in one of the UK trials accidentally received a half dose of vaccine as their first dose in a 2-dose series. And what it turned out is that group may have been more protected than people who got the full dose. And the idea is maybe that they were seeing less development of antibodies to the vaccine virus, not to the spike protein, and that would lead to an improved memory response with the second dose. So that's kind of interesting. They are going to be studying that further. It may just turn out to be a happy accident. There's a relatively small number of people in these studies who had severe illness, but it does appear that after the second dose, there were no hospitalizations and no severe COVID-19 infections in the vaccine group after that second dose, compared to 10 in the control group. Relatively small numbers, but at least promising to note. Just so this graph here on the right is clear, the control group is the quadrivalent meningococcal vaccine. Everyone got some active product in these studies. So what about the variants? So the good news is that the UK variant, which as Dr. Jamil described, is spreading rapidly in the United States, that the AstraZeneca vaccine appears to be pretty protective against it. Now, this is just available in a preprint, but it's freely available online. It's been submitted to the Lancet and is under review right now. However, it appears also to be dramatically less effective against the B.1.351 variant. Now, that is, again, just off of a preprint, and it's important to attach some caveats to that. What they saw was a 10.4% reduction in mild to moderate disease in a young, generally healthy population against the South Africa variant in a trial conducted in South Africa. So what does that mean? We don't know. Probably it has reduced efficacy against severe disease, but how much, we don't know. So there's really no data yet released on what is the effect in older adults. What is the effect in higher risk patients? And is there still a reduction against severe disease? Because ultimately, if you are preventing severe disease, then it becomes a little less important to not have much of an impact on mild to moderate disease, assuming that you have a broadly vaccinated population, because obviously those people with mild to moderate disease can still transmit to an unvaccinated host. So this is a challenge. I will mention parenthetically that Astra has published some initial data indicating that their vaccine reduces the risk of transmission by up to about 75%. And so that is meaningful and good to know. There is some very soft evidence coming out of Israel that the same is true with the Pfizer vaccine as well. Next is a question of dose. This is a 2-dose vaccine. And as I mentioned earlier, there is this idea that maybe a low dose, we'd call a prime boost strategy, with a low initial dose followed by a larger secondary dose, could be a superior option. All right, now how about the Johnson & Johnson vaccine? So the Johnson & Johnson vaccine is being made in collaboration with their subsidiary in Belgium, which is Janssen. So Johnson & Johnson and Janssen are using a very similar design as the Oxford vaccine, except they use a human adenovirus 26 as their viral vector. So right now, really all that's been published is the interim results of a phase 1, phase 2A trial. And what they showed was that you have with both a high dose and a low dose, and I'm only showing the older age cohorts. They did similar studies in younger adults as well. But what you see here is that there is a vigorous antibody response. The far right is a human convalescent sera. So comparing the antibody levels elicited by their vaccine to people who have recovered recently from COVID-19. And you get levels comparable to those who've had natural infection. So that's certainly encouraging. They also measured Th1 responses, so cell-mediated immunity, and they found, again, consistently elevated levels of Th1 response. And they measured that by measuring IL-2 and interferon gamma levels after exposing patient blood specimens to SARS-CoV-2 antigen. So again, encouraging in vitro data, but how does this play out in protection? As I discussed earlier, we are practicing a certain degree of medicine by press release right now. So they put out a press release about 2 weeks ago. So far, I haven't been able to find a preprint for this, but what they did describe is an overall 72% effective against all infections in the United States and 66% effective overall in preventing moderate to severe disease 28 days after vaccination. In the US, what they found is 85% effectiveness overall preventing severe disease, which is great. And they also noted protection against multiple variants, specifically against the B1351 lineage. So that is certainly very encouraging. They did do some of their trial in South Africa, and we're specifically looking for this lineage in the study. So good news, but again, the Johnson & Johnson media relations website is not, to the best of my knowledge, the indexed literature. So I think we'll need to get a lot more data before we can be informed about this. There are some very real advantages to the Johnson & Johnson vaccine, specifically that it is a single dose vaccine, and that is huge. All right, how about protein vaccines? So again, these are more kind of traditional vaccines, like say the hepatitis A vaccine, where you have a subunit that then is injected into the patient, taken up, elicits usually an immune response. What's interesting about the vaccines under study here is that they are combined with adjuvants that allow for adjuvants and delivery vehicles that allow it to be taken up intracellularly and will actually induce a degree of a cell-mediated response despite being purely subunit vaccines. So again, a way around that immunologic problem. So the Novavax one is one that's furthest along. That is a recombinant nanoparticle composed of trimeric full-length spike proteins. It has a saponin-based adjuvant called Matrix M, which sounds awesome. They right now have just published phase 1 data, and I've circled the relevant part of their phase 1 data. This is looking at microneutralization against SARS-CoV-2 in vitro from patient sera, and it shows that both the low dose, the 5 microgram, and the high dose, 25 microgram doses in combination with their adjuvant were highly immunogenic, and immunogenic in a way that again compares with human convalescent serum. So very promising, but of course we have to see how this actually plays out in practice. Again, press releases, waiting for even a preprint, but these things move fast. They are reporting 89.3% efficacy in a UK phase 3 trial. And they also report clinical efficacy against both the UK and South Africa variants. Now, we will need to see this in greater detail to find out really what that meant, but nonetheless, very encouraging. We're hoping to hear more kind of formal peer-reviewed data from the Novavax vaccine soon. Now, this is great, right? The variants are a concern. And I think, I think most of us in ID who've done any degree of virology research, we are not surprised that RNA viruses mutate. That's what RNA viruses do. They mutate, they change over time. So this is in no way, shape, or form a surprise. The fact that there is some attenuated protection is also not a surprise, although I suspect that we will still see some residual protection. And really one of the beauties of these platforms, particularly the mRNA vaccines, is how readily they can be adapted to reflect these new variants, right? That you just, you go into the code, you swap out a couple C's for G's in the mRNA sequence, and you have a new vaccine. And it's worth remembering that in our careers, pneumococcal vaccines, for example, have undergone changes to reflect new strains and new types. The meningococcal vaccine, the same thing, that this is actually a fairly common issue in vaccinology. It's simply that the acuity of the SARS-CoV-2 pandemic has made it, obviously much more urgent than it usually is. That being said, this was a report from Kaiser Health News. It says nearly half of American adults now want the COVID vaccine, then another 30% said they want to wait till the vaccine has been available for a while to see how it's working. This is generally quite encouraging. A relatively small number of US adults are completely resistant to getting the COVID vaccine. It's about 20%, right? So this is good news. However, this is looking at disparities. So communities that have been hit the hardest by COVID-19, people of Black descent, people of Latino descent, 51% of white adults know someone who has gotten the COVID vaccine or has been vaccinated, him or herself. whereas only 38% or 37% of people of African American or Latino descent have the same. So the groups that are at the greatest risk of having severe COVID are also the groups who are least likely to be getting the vaccine. We've seen this in San Diego County, where sites set up in largely Latino or largely African American communities by our county for vaccination, often people from more prosperous parts of our county are signing up for vaccinations at those sites. This was obviously entirely inadvertent on the part of the county, but our attempts to reach the communities in the greatest need needs to be redoubled and redoubled again and redoubled again. These are our real barriers, right? The vaccines will need to be modified to reflect genetic changes, but more importantly, getting the vaccines into the people at the greatest risk. That's our greatest burden. Just a quick summary of the efficacy of the vaccines overall. Pfizer vaccine, 95%. All of them appear to be okay against the UK variant, maybe a little less for Pfizer and Moderna against South Africa. The AstraZeneca vaccine is somewhere between 70 to 90% effective. Not so hot so for the South African variants right now, whereas Novavax and Johnson & Johnson, a little less than Pfizer and Moderna, but still pretty good, and do appear to have preserved activity against the South African variant. So our biggest barrier is uptake, right? No vaccine works when it is still in the syringe or it's still in the vial. We do have a great vaccine pipeline. It is robust. Expected viral evolution is unlikely to foil vaccine efficacy completely, but modifications to optimum protection will probably be necessary. I have to acknowledge a number of people. My good friend, Colonel Julie Ake, who I borrowed a few of these slides from, and her colleague, Dr. Kayvan Mojarrad, both at the Walter Reed Army Institute of Research, along with their colleagues, Dr. Rowland, Dr. Joyce, Colonel Nelson Michael, Dr. Nelson Robb, Dr. Bassan, Colonel Tayan, Colonel Jess Cowden, and Dr. Matt Hepburn. Thank you all again for the opportunity to speak to you today. If you have any questions or concerns, shoot me an email, and thank you very much.
Thank you, Dr. Maves. That was excellent, and you've kept us all riveted, and we really appreciate your time this evening.
Thank you for the opportunity.
I'd like to segue to Dr. Charles Dela Cruz from Yale. And please, Ryan and Shazia, excellent presentation. Please stay on for some, or if people have questions, respond to them in the chat so we can also listen to Dr. Dela Cruz. So I'd like to introduce Dr. Charles Dela Cruz, who is calling in from the East Coast. He's a associate professor of medicine and director of the Center for Pulmonary Infection and Research at Yale. And without further ado, the floor is yours, Charles.
Thank you very much for the invitation to speak to the group in California. I'll be speaking a little bit about monoclonal antibodies and some of the insights with regards to the variants. I don't have any disclosure relevant to this talk. However, I'm a PI of one of the oral antiviral medication in a clinical trial that we're doing here in the East Coast. So you've heard nicely about the vaccination strategy from Dr. Maves. Today I'm going to talk a little bit about passive immunity, specifically artificial passive immunity in a form of monoclonal antibodies. One of the first thing that was recently published last year was a work led by the group in Cedars-Sinai, and this is the Eli Lilly antibody product, which is for outpatient therapy, mild to moderate disease. Dr. Jamil nicely described the spike protein on the virus, and specifically the receptor binding domain in red is quite important for its activity and binding to the ACE2 receptor of the host cell for its entry and infection. Many of these monoclonal antibodies are specifically targeted towards this RBD region to prevent host entry. So this study was initially a phase 2 study, outpatient, was published sort of halfway. It looked at different dosing. It's a one-time infusion. Specifically, a majority of these patients had at least one risk factors for severe disease, elderly, more BMI that is more than 35 and at least one coexisting illness. Interestingly, most of these patients had a median days of symptoms around 4 days when they received the infusion. And the primary outcome of all these studies is essentially our change of the baseline viral load compared to day 11. And so one thing to note is that in these studies, you can see that the mean PCR cycle threshold are actually quite low, meaning that their level of virus is quite high. And then that makes sense based on the life cycle of the virus in an infected patients, because these patients were enrolled quite early, 4 days into their symptom onset. And the study result showed that in the median dose, 2,800 milligram of bamlanivimab showed decrease in viral load that is statistically significant. And then there were much smaller differences with the lower or higher dosing in this group of studies. What's interesting is that these patients using these IV infusion had lower severity of symptoms compared to placebo. And then more importantly, although this was not the primary outcome, COVID-related hospitalization was significantly lower compared to placebo. This antibody therapy actually has fairly good safety profile. Most patients have fairly mild symptoms, and it was no different compared to placebo. So then this study came around, which is earlier this year, which was the completed study of what I just presented in JAMA. And essentially, this study extended the study with a combination of two different antibodies against different parts of the spike protein. And then compared to 400 plus patients, this had additionally 150 more patients in this completed study from the one I presented from New England Journal. The primary outcome here is a change from baseline viral load at day 11. And the results from this full study and a completed study was a little bit different. It showed that the viral load change from baseline compared to day 11 was mostly significant for the combination treatment. And as you can see here and above this graph, it's the blue line that you can see that is lower. And so lower here meaning higher CT value of 35, meaning lower viral load. But then the other single dosing for the antibodies at all these different other doses did not show statistically significant decrease in the viral load, which is a little bit different from what was presented in the interim. Some of the suggestions was that this is because many of the placebos was followed further along, and then it suggested that a combination therapy at least for this study was much better than the single monoclonal antibody treatment. What I thought is important, though, is that ultimately you care about how did the patients do, whether in terms of hospitalization or ED visits. And you can see roughly 6% of the placebo ended up requiring some attention. But then the individuals who received either a single dosing or a combination dosing had a much better outcome in terms of progression of their disease. Their conclusion here that treatment using the combination was better than the single IV infusion alone. I think in the setting of variants and concerns of mutants, I think the combination therapy sort of makes sense. However, in these studies, they did not look as specifically at these variants in the patients that were infected. And so Eli Lilly has done additional passive immune antibody studies. One was part of the NIH Active 3 trial, which is our hospitalized patients that included remdesivir. This was actually stopped in the middle of October, mostly because of after the analysis in DSMB, it did not show major benefit for the patients who received the IV infusion. And so it was not recommended for IV infusion antibodies for people who are already hospitalized. There's an ongoing study currently in phase 3 study, single monoclonal antibody for the prevention of COVID-19 in residents and long-term care facilities. So the other product, which is the Regeneron product, was similarly published in the fall of last year. This is a randomized control trial, phase 1 to 3 study on non-hospitalized patients. This study included two different types of monoclonal antibody that's named here. They had roughly a study that's 1:1:1 based on the different dosing versus placebo. Again, it's a one-time infusion. Median age around 40. Really good representation of male and female. However, I think you can see the diversity is in terms of mostly the white Caucasian population was represented. Again, here, the median days of symptoms for most of these patients are quite early. If you remember, the other one was 4 days. This is 3%. In this study, they actually looked at patients whether they were seropositive or seronegative in the beginning before the transfusion, which is kind of interesting. And then their endpoints is looking at day 1 versus day 7 in terms of viral load, and as well as looking at how many of them ended up with a medical visit at the end of their study at day 29. And so serologically, the status of these individuals correlated with their baseline viral load, which makes sense, right? So if people who have high baseline serologies, they tend to have lower viral load or vice versa, suggesting that these antibodies, even from a natural setting from the infection, actually is helping to clear the virus. In the placebo arm, patients who are seropositive end up having symptoms improvement much faster than patients who were seronegative based on their initial infection by itself. This is even before antibody treatment. And so the results for this group show that the combined dosing of these antibodies have significant decrease in viral load at day 7. This is specifically more important for people who have antibodies that were seronegative at baseline. But overall, even those that did not have, that did have some level of serum serologies, they actually showed overall decrease in the virus. And clinically, sort of like the other studies that I showed, the patients who were given IV treatment were actually had less visit to the for medical care, hospitalization, or ED visits. And this is much more impressive, even more for patients who have no serum antibodies at baseline. And so in terms of adverse reactions for both groups, in terms of significant or severe adverse events, it's actually quite low. There's been no reports of death, mostly infusion related and some hypersensitivity reaction. So relatively very safe therapy for these patients. So this is a bioarchive, as what you heard from other speakers. A lot of the data is really in preprints and also or in press by our media. And this is a group from Dr. David Ho's group in Colombia. And what they've done is they've looked at actually the resistance of the Devereaux variants, one from UK, the B117, and the one from South Africa, the B1351. And really looking at the RBD, receptor binding domain, antibodies that were directed to that region versus the N-terminal region. Here, as you can see in the upper graph, the one in peach color is the N-terminal domain versus the RBD ones are in green. And you can see that if the antibodies are targeted towards the N-terminal areas of the spike protein, you can see significant resistance of the ineffectiveness of the monoclonal antibodies. The ones that were targeted, thankfully, against the RBD regions, including the LY code 555, which is the Eli one, and then the Regeneron one, you can see in here, they tend to do well with the UK strain, but not necessarily with the South African strain, the B1351. So suggesting that I think possibly combinations of some of these antibodies may be important for sort of future strategies. So last year, with some of these earlier reports, emergency use access was given for criteria for outpatient monoclonal antibody therapies. This is some updates from some of the criteria based on people with significant risk factors, including age, individuals with comorbidities, or patients who are younger, and as listed here. Interestingly, the IDSA and NIH haven't fully embraced monoclonal antibodies. I think in end of January, what they suggest against the use of, say, bamlanivimab, strongly against the use for an inpatient, given some of the ACTIV-3 trial study, and an unconditional recommendation against the routine use for outpatient. I think most of this is, is for emergency use only, and same thing for the combination from Regeneron. We have at Yale been using the emergency use access criteria for patients using similar criteria. I think there's some tweaking that we use for patients in outpatient, because since last fall, there weren't that many options for patients who are in the outpatient setting with severe disease, but not necessarily meet the criteria for hospitalization. We've given probably near 400 of these IV infusions for patients, and they are well tolerated. I think we only have a very, very small handful of patients ended up progressing. And so really, I think the study suggested that it does help these outpatient with severe risk factors. In conclusion, I think, just for time, I think the use of monoclonal antibodies for these high-risk patients are quite promising. I think the strategies that you've heard, the same for vaccines and sort of antibody epitopes, is really to find out where these antibodies are targeted. On the right, people are starting to work on specific areas of the spike proteins to look for areas where total escape mutants could come from. You can see examples of the Regeneron and also the Eli antibodies where it can target, and the ones in, say, gray and the white areas are areas where actually it's quite important for the virus to have not mutate. Otherwise, the virus won't be viable. So people are really targeting in the red zone here to find out antibodies that would fully neutralize the virus and also prevent escape mutants. I think part of the strategies that probably could be working is the cocktail of antibodies. I know different companies are now combining their products to develop studies for combination therapies. And what's good about, I guess, these monoclonal antibodies are they're relatively safe so far in terms of monitoring for their adverse responses. So thank you very much. So this is me getting my vaccines from the VA. I think it's great to talk to you, our colleagues in the West Coast, and as we sort of similarly try to fight this pandemic. Thank you very much.
Dr. De La Cruz, thank you very much. That was excellent, and I appreciate your time, most importantly because it's late at night for you. So thanks for spending a Friday night hanging out with us here on the West Coast. And it just shows the endurance of this group. You know, we started with 75, we're in the high 60s, despite going over by 20 minutes on a Friday night. So that just shows the quality of everybody. So Asha, what do you think we have time for a few questions? Do you want to just leave it open for a little bit? What are you thinking?
I think if anybody has any questions, please feel free to open your mic and ask. I know this is an area of probably more questions than answers, but this group is highly engaged. So we want the forum, this to be an opportunity for all of you. Well, I'll go ahead and I had posed a question, and thank you, Ryan, for answering that regarding pregnancy. We're starting to get a lot of questions about pregnancy as well as monoclonal use and the antibodies. So thank you for answering that. The question was about, I have a 6-month gestation woman who came from Mexico, got COVID twice, proven, swabbed twice, symptomatic last year, and really wants the vaccine. I'm just worried the reactogenicity of that and maybe preterm labor, but Ryan, if you want to comment on pregnancy and the vaccine.
I mean, it's an unknown, right? There have been probably about 1,000 women who are pregnant who are documented as having received the vaccine between some of the mRNA vaccines kind of taken together, between some of the international trials and some registry data. And so far, it seems to be relatively safe. Now, Obviously a first trimester pregnancy and a third trimester pregnancy are different. I will say the reactogenicity, I've observed the same kind of anecdotal thing that you have, that it seems like folks who've had COVID may be a little more reactogenic, but I don't know. I mean, I don't know if that actually plays out in practice. I mean, we remember the ones that are, but I'll just, to a counter argument, is my wife had fairly significant outpatient COVID some months ago, recovered nicely, and had a complete non-event with her receipt of Pfizer for her work at Kaiser. So, I think it's a bit of a crapshoot there, but just like we would for everybody, we just have to counsel them, right? I mean, I certainly would. I think the risk to her of reactogenicity is less than the risk to her of COVID. And that's kind of what I think about vaccines all the time, and I think we've all, probably everyone on this call, has had some pregnancy associated catastrophe with COVID. I just, wasn't that long ago, I was calling the team at La Jolla down with Denny to Scripps and Chula to cannulate a pregnant lady who I'd spent all night not quite coding. And I think that I would like to never do that again.
No,
I think that's the key. Many of you may have seen this one letter that came out in JAMA today, actually, from the University of Washington, where they followed up outpatient symptomatic COVID, and still almost half of those patients had symptoms now at 8 to 9 months out. So I would much rather get the vaccine and take my gambling chances, regardless of pregnancy or immunosuppression. And that's at least what we're telling our patients here. We do know there's going to be long-term symptoms and sequelae with COVID. We don't have with the vaccine, and I can't think of a single vaccine that does have that. So at least off the top of my head. So I agree.
I think about the Shingrix, the subunit shingles vaccine that predictably makes people feel like they were punched in the gut, and my outpatients line up for that. I would argue that the mRNA vaccines are no more reactogenic than those, and I think that's an acceptable risk.
Ryan, it's good to see you. The last time I saw you, we were sharing a drink in New Orleans, as I remember.
Yes, indeed, or maybe it was San Antonio, somewhere.
San Antonio, that's right. Yeah.
It's good to see you, Tim.
So what type of reactogenicity are you worried about? I mean, certainly they're not going to have any higher risk for PEG, anaphylaxis or something like that. Are you concerned about some kind of life-threatening reaction, or
no, just general vaccine reactogenicity, febrile reaction, headache, malaise, something lasting a day or two. Worried is not the word that I would use, but I think we got to warn people, right? I mean, there was a very nice opinion piece in JAMA Internal Medicine right before the EUA rollout, where a nurse investigator at, I want to say UCSF, described her experiences volunteering for, I want to say, the Moderna trial. And she said how she got her vaccine, and the next day she, was completely wiped and felt miserable for a few days, and then got better, and said, and she made two great points. One is that we in clinical medicine have to be candid with patients about the reactogenicity, so they're not scared, they're not surprised, but it's worth it. And she said, and I would do it all again. And that's the lesson. I was lucky. I had no real side effects, but a number of my partners, including some of our mutual friends, Dr. Morris, were pretty wiped out, but they got better and they were fine after a day or two. And I think as long as we are honest with patients about that, not saying, oh, it's no big deal, but yeah, no, there's a pretty good chance you're going to feel like crap tomorrow, but the day after that, you'll be great. And your risk of COVID is dramatically reduced. That's all. I'm not worried is not a word I would use.
To be fair, those Navy colleagues are pretty wiped out the night after we.
No, it's true. I told a couple of my Navy colleagues who had some bad side effects. I said, no, side effects are common among, children, weaklings. Obviously, that's not true. The toughest person I know, who is Denny's colleague, my colleague, Charlotte Thomas, was pretty wiped out, if I recall, as well. And Charlotte can spit bullets and fight off elephants with her bare hands. So there's no correlation, unfortunately. As funny as it was the day it happened.
Well, thank you. Thank you to this excellent panel and everybody who's participated on this session. It was recorded, and it will be posted on the CTS website for everybody to review or share. I think this is definitely one of those that we'll probably need to review again. And so thank you all for this excellent presentation, Charles, Shazia, Ryan. Our next meeting will be next week, and we had done this around surge, and I think we need a debrief. I want everybody to come with some lessons or some things that they wish had gone better at their institution, their office. We just dump it out there, and hopefully we'll realize we're all not alone in this. And then we're going to segue in March. We're going to toss the ball to Shazia and the CTS Board and Education Committee to continue these sessions, but probably every other week, so that everyone can have some quiet downtime. We really appreciate all of you, and the feedback has been tremendous, and it's been uniquely California focused, but we've received help from our East Coast colleagues, so we really appreciate that, because otherwise we can get a little tunnel vision. I again thank you all for your time, and if anyone's interested, get online and join CTS as well. With that, Christian, any last thoughts?
No, I just want to thank you. You've really been great, Asha, in pulling all these awesome speakers together and taking the leadership. So thank you. You really make us all look good. So thanks.
Well, CTS, I have to admit, has some tremendous brain power, and everybody in this room and in this chat group and the participant group. It's just an honor to be a part of this state and the community. And so thank you all. With that, have a great weekend.
Bye, everyone. Thank you.
Thank you, everyone.
Bye-bye.
Bye-bye.